Good morning, grade 10. Today our lesson is about identifying DNA as a genetic material. Now, we learned before about Mendel's findings about heredity, Morgan, and other scientists. Okay? They told us about chromosomes, but none of them tell us or discovered about the DNA. So, in this lesson, we will learn how the scientists make experiments to identify that DNA is a genetic material. First scientist is Griffith. Griffith found a transforming principle. He was thinking about what is the genetic material of the organisms. So he made experiment using bacteria called pneumonia bacteria. They cause pneumonia. He used two forms. One form was pathogenic, yani deadly, harmful, and the other strain was non-pathogenic, which means not deadly, not harmful, not pathogenic. So they used different mice. He injected the first mouse with the pathogenic bacteria, and the mouse, as a result, dead. Then he injected the R strain, which is non-pathogenic, in another mouse, and the mouse still alive. After this, he take the strain of S and he killed the S bacteria by heat. Then he mixed it with the R bacteria and inject them together into another mouse. He found a surprising result, which was that the mouse dead. Okay? He was thinking about what's happening. He concluded that there was a transforming material that passed from the dead strains S to R and make them deadly. So R is transformed. It was not deadly and it becomes deadly. All right? But he didn't know what is this transforming material. He found out that there is a transforming material, and they called this studying was transforming principle. After Griffith, Avery and his colleagues, they make experiments to identify this transforming material or transforming principle. After their experiments, they concluded that this transforming principle is DNA. What did they do? They take the S strain, okay, harmful, and they separated the components, each component alone. They use component A and they mix it with R strain, which is non-pathogenic, and they inject it in mouse. Then they take component B, C, and D, and the same thing. They mix it with the R strain, which is non-pathogenic bacteria, and inject it into mouse. All the mice still alive except one. It was dead. So they take the component that kills the mouse and they make experiment about this component. They found that this material would be DNA. They make qualitative tests, they make chemical tests to see that the makeup of this material was the DNA. And they use enzymes also to make sure that this material is DNA. So from their experiment, they concluded that one component of the S strain caused the R to be transformed and kill the mouse. Now they tell that DNA would be the genetic material, not a protein, because scientists assumes before that protein is the genetic material. But after this experiment, they said or they found out that DNA is a genetic material, but they don't confirm their findings, their conclusion. They don't confirm it. After their studying, there was another study did by Hershey and Chase. This study confirmed that DNA is a genetic material. We will talk about it tomorrow. Take care, guys. See you.